Hello, everybody. So it is 12 o'clock and we are going to go ahead and get started. Thank you so much for joining us today. This is the last of 5 webinars that we hosted this season. We'll be announcing the topics of our fall webinar series this summer, so be on the lookout for links to register for that. They'll be coming up in our Go Botanical newsletters. If you aren't already registered for Go Botanical and you'd like to be, I'll be dropping the link to that in the chat box here in a moment. This webinar is being recorded. We'll be sending the recording out to you sometime in the next few days. Please feel free throughout this presentation to ask questions using the Q&A feature or drop them directly into the chat box. We'll be holding a general Q&A at the end as well. We'll try to get to all of your questions, but if we don't, feel free to follow up with me via email, which I'll drop into the chat in a moment too. We're hosting today's session from our Botanical Research Center on Kauai. I have with me Dr. Nina Ronstedt, our Director of Science and Conservation, who will now tell you a bit about today's session.
Thank you so much, Amanda, and welcome to all of you, and thanks for joining us. So we've created these webinars to give you all a chance to meet some of the scientists and conservationists behind NTBG's many different science and conservation products. And today's theme is about plants that occur along streams and coastlines. And we created this theme because water is a really critical part of any ecosystem, and especially here on islands where fresh water is really what our life depends on, and also plants help control erosion from both mountains and the coast. And it's a really dedicated system or delicate system that needs to be in balance for all life to thrive and to be resilient to big weather events and changes from thunderstorms and excessive rainfall to increasing tide lines. Some of these events are things we are expecting to see more of in the coming years. So we have a really nice little series of talks for you today. First, you will meet Puakea Mo'okini-Oliveira, who will be taking you to NTBG's Limahuli Garden and Preserves on the north coast of Kauai and tell us about restoration of the core Limahuli stream there in order to improve the water quality for native fish. And after that, Seana Walsh will be telling you about her work on studying salinity tolerance of coastal plants so that we can better understand which ones to use in conservation projects and also get a general idea about how our native plants will be doing if we will have increasing salinity under potential climate change scenario here. So we hope you will enjoy it. And with that, I'll give the word back to Amanda. And thanks again for coming on.
Thank you, Nina. So Puakea is going to share her screen here right now for us, and we'll go ahead and jump into the first session. And like I mentioned, please feel free to drop your questions into the chat box. Uma from Limahuli Garden is here as well, and she'll help us answer those throughout. All right, take it away.
Okay, aloha kako, aloha all, and mahalo for tuning into this webinar. Like Nina said, my name is Puakea Mo'okini-Oliveira, and I'm so excited to be here with you folks today sharing this presentation titled Lessons from the Stream, the Ha'aina Oʻopu Restoration Project. I've been with the National Tropical Botanical Garden, specifically at Limahuli Garden and Preserve on Kauai since 2019 as a conservation technician. And just a few weeks ago, I wrapped up my first year as a Master's of Environmental Management student and Ha'oli Maloa Foundation Fellow at the University of Hawaii at Manoa. And I mention this because this project and the stream itself and the people that I've met along the way are a lot of the reason what inspired me to pursue my graduate studies. So I'd first like to thank all of the contributors, both in the planning and developing stages, and of course, those directly involved with carrying out the field work. And a special thank you to the funders of the project, the Hawaii Fish Habitat Partnership, part of the U.S. Fish and Wildlife Service. So the remote Hawaiian archipelago was birthed from volcanic origins in the middle of the Pacific Ocean, which became home to our Polynesian voyagers and ancestors thousands of years ago. And what many often do not realize is that the Hawaiian archipelago is comprised of some 137 islands, including islets and atolls. And our location has resulted in having a high degree of endemism, meaning we have species that are only found here in Hawaii and sometimes in specific locations. Of the 8 high islands, only 6 are accessible to the public, but that does not stop the masses from coming here to experience Hawaii's unbeatable beaches, majestic mountains, and rain and wind-carved valleys. And while we boast some of the most unique and wondrous landscapes, seascapes, flora, and fauna, it has been at a price. So the Hawaiian Islands have been experiencing extinction pre-contact, but it weren't until recently that we've been dubbed the title extinction capital of the world. Native and endemic marine life, avifauna, insects, mollusks, and plants are clinging on to survival. So of course, at the botanical gardens, we're extremely focused on those endangered, threatened, and vulnerable plant species. But as any good ecologist and optimist would have you believe, it's all important. Rather, of course, all of our ecosystems are connected. So we kept this holistic ecosystems approach in mind throughout the Ha'ena Opu Restoration Project. And today I've been asked to speak on some of the riparian restoration work that took place. But I first wanted to give an orientation to our project location as well as some of the broader components of the study. So our project took place in an ahupua'a named Ha'ena, and an ahupua'a is a social ecological community that is separated from others with distinct geographical boundaries. Before the idea of private property was introduced in Hawaii, ahupua'a were cared for by commoners and looked over by appointed ali'i, or lower-ranking chiefs called konohiki. And many ahupua'a extend from the top of a mountain or ridge line down into the valley and towards the nearshore fishery and into deeper waters. So many have likened them to watersheds or ridge to reef type namings, but not all ahupua'a follow watershed boundaries depending on location and island. So Ha'ena, where this project happened, is regarded as a wahipana, or storied place of old, and is referenced in numerous cultural traditions and stories. So residing in Ha'ena is the verdant valley of Limahuli, which you see here, which translates to turn one's hands. Limahuli Valley is currently stewarded by the National Tropical Botanical Garden, and they uphold a paramount role as a refugia for many endangered and endemic flora and fauna in living collections, and strives to serve as a foundation for education for the broader community. So evidenced by the agricultural complexes throughout the valley, the original caretakers of Limahuli would have actively managed the riparian areas or areas adjacent to the stream to maintain their auwai, or irrigation systems that fed the production of the staple starch kalo or taro directly, which is what you see on the left image there. So by the mid to late 20th century, Limahuli saw the increase of invasive species such as wild cattle. And while there are a number of harmful effects caused by unwanted plants and animals, we highlight that the presence of heavily shading trees may be driving a more diatom-dominated aquatic environment in Limahuli stream. So by the Hawaii Stream Index of Biological Integrity, Limahuli Stream was scored as excellent, exhibiting near pristine conditions. And with our efforts, we strive to keep it that way. So by strategically managing the stream, we can ensure that the direct effect on aquatic ecosystem function and resilience for downstream users is a healthy one. So why Oʻopu, or what are Oʻopu in general? So Oʻopu are the native fish of the goby family, and there are 4 freshwater gobiidae, and the 5th is in the genus Elotrius. And so one of the adaptations that make Oʻopu so cool, both to myself and of course to the kids, is their fused pelvic fins. So this allows the Oʻopu to climb up the stream with their fused pelvic fin kind of acting like a suction cup, allowing them to stay on rocks or be under fast flowing water. Of course, being isolated in the Pacific Ocean, it's not too big of a surprise that there are only few native freshwater aquatic species, and some are primary consumers and some are secondary consumers. However, all Oʻopu consume algae, herein referred to as limu. So the Oʻopu live an amphidromous life cycle, and they're signaled by freshets and seasonal floods. Their eggs are carried downstream into the nearshore fishery, where they drift with currents and develop in their planktonic state. During the next set of freshets, around 6 months later, the post larvae, what we call hinana in Hawaiian, are signaled by those freshwater inputs to begin their journey back into the stream. So because of this, Oʻopu can be directly affected by the alteration of aquatic ecosystems. And as a result of alterations such as channelization and stream diversions, many of us born in Hawaii may never have heard of, seen, or much less eaten an Oʻopu. The Ha'ena Opu Restoration Project aims to preserve and enhance overall watershed health with a focus on the aquatic species and the riparian areas in which they're intricately tied. So we sought to do this by increasing the light levels along 1,500 feet of Limahuli stream and replacing the invasive species with appropriate native riparian plants. Additionally, we also monitored canopy and bank vegetation changes, Oʻopu and other aquatic species abundance, as well as measure the change of green algae. These are our project goals and the specific questions during driving our methods. Can we initiate a regime shift from a diatom dominated to a chlorophyte dominated environment by illuminating the stream corridor during canopy removal? And does that effectively increase Oʻopu abundance within our restoration sites? The project occurred within the lower Limahuli Preserve, and that's separated from the upper Limahuli Preserve by an 800-foot hanging waterfall. And the headwaters are around 3,000 feet in elevation. But Limahuli Stream is a narrow second-order perennial stream, meaning it flows all year long, and it's just under 2 miles from the waterfall to the estuary. So the first step of the project was to remove selected invasive trees that created densely shaded canopy over Limahuli Stream, which may inhibit the growth of limu, the green algae, the integral linkage of the aquatic ecosystem for the Oʻopu. The tree trimming began in the fall of 2019 by Halelea Tree Service and a few NTBG Malama'aina technicians. And then restoration, including native outplanting and monitoring efforts, continued until September 2021. So guided by previous restoration managers and NTBG living collections experts, we selected fast-growing hardy plants with strong root systems to prevent future erosion, with an emphasis on housing genetic reservoirs of Northwest Kauai region specific species whenever possible. And so this is an example of one of our restoration sites named Reach 2. And it's one of the first sites our crew got to work on. And it's one of my favorite examples of ongoing restoration progress. By September 2021, we were able to successfully outplant over 1,600 suitable trees and shrubs. And they've been supported by over 3,000 ground covers. So keep in mind that this restoration area is only accessible by foot, so all tree clearing equipment, outplants, and site maintenance tools are carried in. So this is Kokio ke'o or Hibiscus waimea subspecies Hanare, and it's an endangered and endemic species that provides an example of some of those key plant characteristics that I mentioned just in the last slide, which our restoration was founded on. And the Kokio ke'o is one of the only native hibiscus with a scented flower. So it can serve as a potential moth pollinating plant. And that picture of the moth there on the right, I've come to learn that it's probably not a native one, but I did find it next to this open blooming flower. So these maps are showing the location of the threatened and endangered species that we've outplanted on the left. And on the right is a summary of our restoration maintenance effort. So some of the first areas that were cleared on the downstream and the lower reaches have had more effort to maintain sites. So that's why some of the circles are a little bit bigger. So these graphs are showing the amount of total basal area of invasive canopy species removed before and after treatment. And the x-axis is showing the treatment time, and the y-axis is showing the total basal area removed by tree species. So total basal area is the cross-sectional area of a tree at diameter breast height around 4.5 meters. And it's often used by timbers and foresters to determine stand density of a tree in an acre. But we calculated our basal area to centimeters squared because we were measuring trees in a 5-meter circular plot on both sides of the bank and in each restoration and control site. So nearly 35,000 centimeters squared of total basal area was removed overall, which was predominantly the Schefflera actinophylla. And interestingly, we saw a decrease of basal area in control sites overall post-restoration, indicating there are potentially numerous tree fall events independent of restoration efforts along the stream corridor. So in this figure, on the bottom right, we looked at canopy species change by reach. So R1 and R5 were our control sites. And regardless, each site displayed a decrease in total basal area over the project period. And again, Schefflera was a notable and the dominant targeted species. So our conservation manager, Uma Nagendra, also ran a principal components analysis of bank vegetation canopy species. So if you aren't familiar with this type of graph, all you need to know is that the closer together the points are, the more similar their species composition is. The ovals are also showing where a certain type of point clusters. So here we see that the restoration reaches have a slightly narrow community composition following restoration compared to the controls showing a slightly wider composition. And the differences between bank species appear to be driven by the Olerites and Pandanus dominated areas versus the Syzygium, Schefflera, and Psidium dominated areas. We also ran a similar analysis for the Oʻopu themselves, and aquatic animal species composition remained consistent across all sites regardless of treatment. Overall, all sites and dates tended to cluster together, and there is no significant differences of Oʻopu composition within the control and restoration sites before and after restoration. So diversity, therefore, did not change over time. However, control and treatment reaches did show different patterns of diversity over time, potentially indicating a shift in seasonal patterns. So this is showing the animal species abundance change within restoration treatment over time. And we did not see no significant change. So these are Oʻopu that we most frequently encounter during our surveys, the Alamo'o, the Nakea, and Nopili. And interestingly, Nopili counts in the control areas were higher than in the restoration areas. And finally, using our assessment data, we also saw that green limu cover did not change significantly. It did change seasonally, but not it did change seasonally, but did not differ significantly. And the restoration reaches did exhibit a brief increase in green limu cover growth immediately following tree trimming, but it was short-lived. So what are these results telling us? But it certainly raises a few more questions. Firstly, has not enough time passed to be seeing results? Secondly, is canopy openness not as important to the overall abundance of green limu and Oʻopu as we thought? So here we recognize that there are many driving factors to consider that have impacts on aquatic environments, the nutrient inputs, the geography and topography, and of course the flashy nature and dynamic nature of streams themselves. And finally is the lack of restoration at the estuary a missing link. So as we know, the estuary plays a fundamental role in Oʻopu recruitment. So lack of restoration and monitoring here could potentially be evidenced by the insignificant increase of Oʻopu abundance upstream. So this lends to the notion that when we manage our streams, we should identify the entire system as a whole. And for our future work, a focus on the larval stages of Oʻopu within Limahuli and nearby streams would be of great importance to the overall story of Oʻopu in Ha'ena. So all things considered, the project has had many successes to date, some of which are highlighted here. And we hope that the Ha'ena Opu Restoration Project will act as a catalyst for continued maintenance and long-term monitoring effort of Limahuli Stream and to provide insights at both the community and academic levels. So here are the sources referenced in this presentation. I'll be happy to answer any questions as time allows. Mahalo nui.
Thank you so much. That was really enjoyable. Appreciate it. So we are going to move on to our second presentation. So this one is from Seana Walsh, and she's going to be sharing about a salinity project that we did. So go ahead and take it away, Seana.
Great. Thanks, Amanda. Aloha, everyone. Thanks for joining. All right. So my name is Seana Walsh. I'm the conservation biologist here at NTBG, based in Kalaheo on Kauai, and I'm pleased to share the work of my colleagues and I of our work investigating the salinity tolerance of Hawaiian coastal plants at the stage of seed germination. This work has been supported by Sea Grant with co-author on this talk, Dr. Casey Barton from the University of Hawaii at Manoa as principal investigator. So let's dive into our talk, titled Impaired Germination in Hawaiian Coastal Plants with Increasing Salinity, Implications for Management in the Face of Climate Change. I'll just start out by saying that plant species that are able to exist in higher saline environments in general have various mechanisms to tolerate salinity. Pictured here is Sesbania tomentosa, a native Hawaiian coastal species that is federally listed as endangered. And this stunning photo was taken by our research biologist, Ken Wood. So those mechanisms include tolerance to osmotic stress, so plants can close their stomatas in order to do that, exclude salts from leaf blades, and tissue tolerance, so by compartmentalizing salts to avoid high concentrations in their cytoplasm. However, salinity tolerance is highly variable across species and life stage of a species. Pictured here is a native but indigenous species of Bacopa. I also just want to point out that coastal dunes in particular are actually freshwater systems. Not all of the species we included in this study are dune specific, but many of them occur in dunes. And so the plant species that occur in dune ecosystems are exposed to salinity above ground from seawater spray and storms. But with sea level rising, this is leading to intrusion of seawater below ground. And climate change projections also generally include less rainfall, which leads to greater accumulation of salts in surface sands. So with projections of sea level rise, increased frequency and intensity of storm surges, and increased drought, coastal plant species in the Hawaiian Islands will experience an increase in salinity exposure. Although some species may be able to tolerate these changes, the salinity tolerance of most Hawaiian coastal plant species and at their different life stages is unknown. And in Hawaii, as many of you may know, coastal ecosystems are very important for a variety of reasons. They provide habitat for native plant and animal species. They provide sustenance and recreation for residents, stabilize shorelines, which protects existing infrastructure, and contribute significantly to Hawaii's tourism economy. And many coastal native Hawaiian plant species are already also threatened and endangered. This photo is just very obviously demonstrating that intersection of our coastlines providing habitat for rare species such as our native Hawaiian monk seal and contributing to our tourism economy. This is on the south shore of Kauai. So understanding how increased salinity will affect Hawaiian coastal plants is important to better predict the range of impacts that climate change is going to have. Lum and Barton investigated salinity tolerance in the common Hawaiian coastal plants Jacomantia sandwichensis and Cytophallix, and they found that both species are salt sensitive and that sensitivity shifts across development with the lowest salinity tolerance observed in seeds and seedlings. So we tested the following in seeds, again, so a very early and vulnerable life stage of the plant in 17 native and 3 non-native species commonly found in coastal areas. We tested the tolerance or sensitivity to varying levels of salinity, how these varying levels of salinity affects the timing of germination. For example, does it delay it? We also tested for the seed's ability to recover from salinity exposure. And the coastal species we ended up including have to do in part with their abundance and access to collect seeds, the timing that they were fruiting, and being able to obtain enough seeds for this study. We needed about 400 seeds per species. We wanted to include non-native species to explore whether they were more or less tolerant to increasing salinity compared to the native species. But in the end, we couldn't include as many due to time constraints. So we collected fresh seeds from many different plants or maternal founders, and the different salinity level treatments were developed using unfiltered, untreated ocean water we collected directly from Lawai Kai on Kauai, pictured here when we were collecting it. And the diluted treatments we diluted with distilled water. The 35 ppt or parts per thousand was again the full seawater, and this is usually somewhere between 35 and 38 ppt around Hawaiian coastal waters near the shore. Seeds were sown in a germination chamber in our seed lab, and germination trials were monitored 2 days per week for 13 weeks, and we monitored so frequently 2 days per week to get at that timing to germination variable. And here are some beautiful images of the seeds of each species that we included in this study. So this is another outcome. And so I'll orient you to this figure for a moment before I start discussing results. Each box represents a species. They're presented in alphabetical order. And I mentioned earlier, we tested salinity tolerance in 17 native and 3 non-native species, but some data were inconclusive. So in the end, we looked at 18 taxa total. 2 of them were non-native, which I have with an asterisk here. And on the y-axis, we have seed germination percentage. And on the x-axis on the bottom, we have our control of distilled water, which is 0 ppt, and our 3 salinity treatments of 10, 20, and 35 ppt. In general, we found impaired germination with increasing salinity, and you can visualize this with the box plots in each square generally going lower as you go from left to right. And I pictured Bacopa earlier. Here's just an example. So seed germination percentage is generally going down with increasing salinity, like a steps going down. That's just the general pattern we're seeing. And 12 of the 18 taxa, or 67%, did not have any seeds germinate in the 35 ppt salinity treatment, so the full undiluted seawater. And I have highlighted those here in yellow. 6 of those 12, or 33% of the 18 taxa, did not have any seeds germinate in even the 20 ppt salinity treatment, and those are highlighted in red. And so here we have each taxon in the same place. The x-axis on the bottom is the same, but on the y-axis here, we have mean germination time in days. And in general, we found that germination slowed down with increasing salinity. So you can visualize this with the box plots in each rectangle, generally going up higher as you go from left to right. So I'll just look at this Bacopa again. So they're generally going higher. And based on our initial trials of seed salinity tolerance, we have been able to see within what range of salinity a species seeds are able to germinate at the same level of the 0 control. So again, these taxa are listed in alphabetical order, and the last column, tolerance, is indicating that range of salinity tolerance that a species has for seed germination. I've highlighted all the taxa or species with a salinity tolerance of less than 10 ppt in red. So of the taxa we looked at, these are the least tolerant to salinity at the developmental stage of seed germination. I've highlighted the taxa with a salinity tolerance somewhere between 10 and 20 in orange, and those with a salinity tolerance somewhere between 20 and 35 are in yellow. The one species that has a salinity tolerance above 35 ppt is highlighted in green. This is the Sesbania tomentosa I pictured earlier. Interestingly, it's a federally listed endangered species, one of our native coastal species. And there are a couple taxa not highlighted for which the data were inconclusive for this, since germination was so low overall. And just to explain this a little more, using one of the species as an example, Dodonea viscosa or a'ali'i. This species is commonly used for restoration projects. And here it's pictured at Polihale here on Kauai. The salinity tolerance for seed germination for this species is somewhere between 10 and 20 ppt. And the basis for this is that there was no statistically significant difference found in germination percentage between the 0 control and 10 ppt, but there was between 0 and 20 ppt. At the end of this initial trial to looking at salinity tolerance, the same seeds were transferred to a recovery trial. So the seeds were rinsed with distilled water and transferred to clean Petri dishes and monitored 2 days per week for at least 4 weeks. And in this figure, we also have each taxon in the box with mean germination percent on the y-axis, and on the x-axis, the salinity treatments. The gray bars are germination after initial exposure to the salinity treatment and control. So I actually just showed you this data some moments ago, but the black bars here are showing recovery germination. So the percentage of remaining seeds that germinated after the seeds used in that initial exposure experiment were rinsed and resown. And our main takeaway here is that recovery germination varied among species. Seeds of some species are unable to recover from salinity exposure, such as those highlighted here in yellow, while some are able to reach percent germination at a similar level as control in the initial exposure experiment, such as those highlighted here. So here's again pointing out that Bacopa, you can see it here in the gray bars that mean germination went down in the initial experiment with increasing salinity, but after the recovery experiment and the seeds were rinsed with fresh water and resown, you see that even from the 35 ppt treatment, they reached similar levels of mean percent germination as initial in the 0 control. So through this research, we were able to identify the most tolerant species and least tolerant species of the ones we examined at the life stage of seed germination to projected increases in salinity exposure due to climate change. And these results on a species by species basis can be used to help inform coastal management decisions by identifying species that would be more likely to have self-sustaining populations in coastal restoration projects, and also sheds light on those species that may require more intensive monitoring in both natural populations and in outplantings. It can help with decisions on what to plant where. For example, more tolerant species closer to the high tide line and those less tolerant planted further inland. This photo was taken last year at Lawaikai on the south shore of Kauai. So the seeds that germinated in our germination trials were brought down to our nursery for propagation and are currently being used to test salinity exposure in a field experiment at the seedling stage. And our 1-year post-planting monitoring will be taking place next month. I also wanted to add that salinity tolerance of these species is being investigated at the other very vulnerable life stage of the plant, at the seedling stage, by PI on this grant that funded this work, Dr. Casey Barton, and a graduate student, Anna McCormick, at the University of Hawaii at Manoa in their greenhouses there. Oops, skipped one. So again, some additional outcomes of this project include those beautiful seed images, but also being able to bank those extra seeds for future restoration, conservation, and research. And mahalo to all of the organizations that assisted us with seed collections and the many volunteers and interns that assisted with the germination trials. And mahalo to NOAA's Seed Grant Program for funding this research. Mahalo to all of you for your attention. And I'm happy to answer any questions you might have now, or feel free to email me.
Thank you so much, Seana. That was great. What an awesome way to end our webinar series this spring. So feel free to drop questions into the chat box, into the Q&A feature. We have plenty of time to answer some of those for you. And we also would love to hear from you about topics you may want to us to focus on in the future as well. So when I send out the recording, I will also include a link to a survey, and you can fill out that survey and let us know what you thought about this series and let us know what you'd like to hear about in the future. We'd really appreciate it. So I know that we had a couple of questions that we answered already in the chat box. I know Uma helped answer a question about rock walls potentially being in a photograph. Puakea, I don't know if you wanted to talk a little bit about the archaeological sites at Limahuli while we wait for our questions to come in.
Sure, yeah. So there are a lot of archaeological sites within the valley, and I think there have been a number of surveys that have happened since like the 60s as well, documenting and mapping them. So we have evidence of not only habitation, but of course, like the lo'i agricultural complexes. And because they're, they need water, they're usually really close to the stream themselves. So throughout our restoration, we're really careful in the placement. So I guess if you think of 1,500 feet along the stream and the amount of plants we're able to outplant, it might not seem that much because we were being careful about the rocks and not to disturb. In the event, just wanting to keep those complexes clean and safe and not disturbing them, but maybe hopefully in the future to really restore and have them in production again would be wonderful as well. So yeah, we were sure not to put things in the way of anything that might disturb those walls.
Thank you so much. We had a question come in from Stormy. First, she says, wonderful presentation. And can you tell us more about some of the plants that didn't do well with salinity tests? Can we plant them in our yards or gardens? So I think this one's for you, Seana.
Sure, yeah, I was just typing mahalo for the, Puakea, you had a great presentation. I'd say that any native species, especially appropriate to the area you live in, so if it's from that similar type of habitat, is great to plant still. Yeah, so interestingly, one of the two non-native species that we ended up including did pretty poorly. So you shouldn't be planting that anyway, the non-native one, the Haulikoa. But yeah, I mean, if you live in a coastal area, maybe if you wanted to see regeneration with your, if the plant was fruiting and seeding, maybe you would reconsider some of these, but these were like right up near high tide lines. And, it's, the experiment was, if that was the case. So I wouldn't think that you were in an area where there's inundation of the ocean water. So I'd say any of these you were interested in would still be fine to plant.
Thanks, Seana. And I just dropped the link to our gardening resources page in the chat as well. So we do have a few plant care guides in there and some lists in the FAQ of plants that grow in Hawaii. So you can check that out. John asked, let's see, large amounts of silt that gets washed onto the reef during storms. I have made some efforts at restoring the stream bank, but there are large sections where it is bare dirt. Is there someone at NTPG that I can consult about this? No takers, no. Yeah, does anybody know of any resources we could send John to? Are there any extension services through U of H or anything like that?
I think that there are some guides online. I think there was some work done in Waimanalo that have a quick list of appropriate stream plants for restoration. But other than that, I think the guys to ask from NTBG would probably be Uncle Mike, Demoda, and some of the living collections experts. I can tell you that familiar with some like plants that have good, strong root systems. And I know like ahuava was something that we used because it's a native, but I know a lot of other projects, if you're not so concerned about native versus non-native, vetiver is also really good because it's fast growing and has strong roots. But yeah, I don't want to assume I'm an expert on that as well. Just learning to, but I hope that answers your question. I'll try to find the link to that guide online.
Thank you. And before you do that, I have one more question for you from, is it Eunice? How big do Oʻopu get? Often removing trees from banks results in a siltation problem, clogs gills, but I can't think of a fish that eats algae from rocks here.
Thank you for the question, Eunice. So depending on the species, Oʻopu can get mature from, only like 6 inches for some is they're large, and others like the Nakea, more common ones, can be up to 10 or 12 inches or more. Some kupuna watalas, they've grown up with Oʻopu with mouths the size of their fist. So if you can just kind of imagine, that's kind of a size reference. But as far as, yeah, their adaptations to eat from algae. So one in particular, like I said, they all are secondary consumers, but one, Nopili, they actually have adaptations to, in their mouths, their teeth, to actually be scraping the algae from rocks. So they normally feed on the filamentous algae. I'm not sure if that answers both of your questions or comments.
Thank you. So I'll jump over to Shauna for this next one. So Corinne says, these seed salinity tests would be great to do with youth. Is there a protocol document for this?
Yeah, we think so too. And so that's part of the project funded by NOAA, and Dr. Casey Barton has developed like the curriculum and the protocols to do this, and she's doing this at some schools on Oahu. So if you email me, I can get you in touch with her or get the materials to connect you. Great. I can share my email here again as well.
Thank you. And then let's see. I'll pop back over to Puakea. So we have another question from Stormy. Do you know more about how the Oʻopu move through the strong running water using their sucker? I'm imagining they inch along on the rocks.
Aloha, Stormy. Yes, that's correct. They use their sucker, but I've also come across some literature that I've looked into the different jaws and teeth structure of each individual species, kind of noting the ways that, yeah, their adaptations allows them to cling on to rocks as well. So not only beneficial for eating, but as you said, like scooting in the water, some of them actually use their jaws and their mouths to help them along the way.
Thank you. So let's see, I think we've caught up with all the questions, but please feel free if you have any last ones to drop them in for us. And, I just want to take a moment, Shauna or Puakea or Nina or Uma, do you have any last parting thoughts or words that you'd like to share with the group about, stream and coastal restoration?
Well, I guess we thought this was a very appropriate subject to take up, as everything is connected, and it kind of aligns both with that we're working on the plants, we're working on the system, that the water is really important, and that even part of the system is also the usefulness of plants, or in this case for helping the stream, or actually of the fish for being food. So it's just important to remember when we work with conservation that every single species or every part of it has a big role, but also the overall balance of the system together is very important. So yeah, we work from top down to bottom up, or the other way around as well. And we look very much forward to continue that in the fall too. Some of the subjects that we might be bringing up include the preparation for the post-2020 global strategy for plant conservation, and what that means to the coordinated programs in Hawaii. And we're probably also going to look more into useful plants, and that is everything from plants that were brought here by early Polynesians, including things like banana and taro, to recent projects that have been looking into some of the medicinal uses and other things. So there's going to be lots of nice things, and we expect to have about 3 webinars this fall, starting back up in September. But you can see that through our social media, or Go Botanical, or whatever way you're following NTVD. So we hope to see some of you back.
Thank you, Nina. Appreciate it. So, mahalo nui again for joining us today. We really love sharing our work with you all. So like I mentioned, I will be following up in an email with the recording and a survey. So please let us know what you thought about today's session. And I'll also be providing some links for just different ways that you can get involved and help support the work that we do here at NTBG. So thank you all again, and a hui hou. Bye, everybody.
Mahalo.